Thanks, Michael, for the introduction. As Michael alluded to earlier, the field of dark druggable genes faces an immense challenge in trying to characterize and understand genes that have very little information and resources to study them. Our center is excited to face this challenge, specifically for ion channels. But why are ion channels so important to study? I decided to highlight just one example where a mutation in one gene called cystic fibrosis transmembrane conductance regulator, or CFTR, causes cystic fibrosis, the most common monogenic ion channel disease. Cystic fibrosis is a rare but life-threatening disease that until recently had absolutely no cure. Fortunately, last year, FDA approved Trikafta as a drug that has shown very promising results in treating patients with cystic fibrosis. And to me, this is a very clear example of why this class of druggable genes needs to be explored further. However, if you look up CFTR on PubMed, the search returns over 11,000 publications associated with this gene. So clearly, everyone has been studying this gene, and it's well known to cause channelopathy. This slide represents a dozen of other channelopathies that are caused by mutations in roughly 50 different ion channel genes. And even though I'm not going to go over every gene and disease on this slide, I would like to point out that these diseases extend well beyond classically thought nervous system, since a lot of ion channels are heavily expressed in the brain. But actually are also relevant for heart, skeletal, kidney, bone, and metabolic health. It has been annotated that there are about 350 ion channel genes that exist in mammals. About 102, or roughly a third, are still considered dark and are on our list that I'm showing on this slide here. And there are at least a dozen known channelopathies, as I just mentioned, and only a small percentage of ion channel genes are associated with those channelopathies. So likely, this is only the tip of the iceberg that we're dealing with, and we will learn a lot more about ion channels and their importance for human health as we continue studying them. If ion channels are so critical for human health, why so many are still dark? And this relates to a broader question of why some genes are historically ignored. And it is now a nationally recognized issue that has to do with imbalanced propensity to focus research on a small fraction of the proteins. This figure came from a +1 paper that combines bibliometrics with second-generation sequencing to define the brain's ignorome, or the set of genes which are highly and selectively expressed in central nervous system, but yet poorly studied in the neuroscience literature. On the x-axis, you see the number of neuroscience-related references in PubMed as of 2013. On the left y-axis, you see the number of genes, while the right y-axis represents the cumulative literature distribution. And it turns out that a subset of only 30 genes on the far right, the last two bars, absorb 2/3 of the total neuroscience literature. So 30 genes, 2/3 of all literature. Now, in contrast, 200 genes to the far left, the first two bars, absorb only 0.1% of the literature, which is pretty striking. And even more interesting, biological properties don't really seem to predict which genes are hot versus ignored. And it doesn't have to do with the number of interacting partners or the number of motifs or other properties. Instead, popularity is best predicted by the date of discovery. And even though this is not the most recent publication, the message, I believe, still holds true. People write grants on genes for which there is already data and information and resources available that allows them to study these genes and make hypotheses. However, if there is no data, there is no resources, you can't really write a grant, so it creates this catch-22 situation. And it's true for all genes, not just ion channels, but GPCRs, kinases, and other classes as well. And if it's true for all genes, I just showed you how ion channels are key and we need to study them in particular. And since those issues touch the whole field of biomedical research, NIH decided to fund DRGCs to study ion channels in particular, in addition to GPCRs and kinases. Since ion channels are so critical for human health, we turn to studying these dark ion channel genes in mice as model organisms. And to do so, we teamed up with the world-class expert Kent Lloyd, who runs an international program called IMPC to knock out and phenotype every single gene in the mammalian genome. And his program already knocked out and phenotyped over 7,000 of genes. And all mice generated at his center are deposited in publicly accessible repositories, and data is freely available and accessible at impc.org. And there's a lot of very interesting and unexpected data that can be found out there. And I encourage everybody who's on this call to mine this site for their favorite genes. Together with Kent, we've been mining this data specifically for dark ion channels. This slide represents some of this data. And it turns out that 318 genes out of 350 ion channel annotated genes are enrolled in COMP IMPC pipeline, which covers 97% of all annotated ion channel genes. Now, I showed you a list of our 102 ion channel genes on IDG list. And out of those, 94 have mouse homologs, which represents roughly a third of ion channel genes. So imagine a third of the whole class of genes is still considered dark. Now, out of those 94, 60 have been successfully made into a knockout mouse, which represents about 64% of all dark ion channel genes. What is even more striking is that 50 out of those were phenotyped, and more than 50% are associated with significant phenotype, which is really, really unexpected. However, the 44% don't show observed phenotype, and that doesn't really mean that there is no phenotype. It merely means that there's no phenotype observed for things they've been looking at. And they, for example, don't really have a capacity to look for lung health. Therefore, these mice may or may not have a lung phenotype. This table demonstrates the number of IDG ion channel knockout lines that demonstrate a significant deviant phenotype in the COMP IMPC pipeline. To a large degree, ion channel phenotypes are expected to be associated with excitable tissues, as I've mentioned before, and thus we see nervous, behavioral, and endocrine phenotypes. But what we also learned is that systems that are not associated with excitable tissues also showed phenotype. Those include growth, hematopoietic, and aging, which was very unexpected and again highlights previously unappreciated roles of ion channels in other tissues besides brain. Together with Kent, we also created a special web page at impc.org that is dedicated to the IDG project, not just ion channels, but GPCRs and kinases as well. You can search for IDG relevant targets directly, and there are a lot of very cool phenotypic data available, and I implore you to dig deeper and look for your favorite genes on this link. And to make it even easier, we included a table that represents phenotypes for IDG relevant genes. This table shows dark gene on y-axis and different phenotypes on x-axis. And what you can appreciate is that some genes are associated with significant phenotypes, and those are shown in orange. However, if there's no phenotype, the well would be highlighted in blue. And if it's not tested, it would be in gray. And if something caught your attention, you can then click on this green link that will allow you to request whatever is available at a time. Either it's a mouse or ES cells directly. We're focusing on mouse models to dig deeper into relevance of ion channels for human diseases, but we're also engaged in a number of in vitro studies. And there is a lot of basic information that we still lack about ion channels. We do know, however, that they can be found on membranes of various organelles, such as plasma membrane, mitochondria, nucleus, ER, Golgi, but we have a very poor understanding on how the localization is regulated or executed in those cells. To address this, we put together a team with Peter Sorger at MIT, who developed protocols that allow us to use cyclic fluorescence to determine ion channel localization, and this will be done using high-throughput confocal microscopy. Another important direction we're taking is to perform functional high-throughput screens to identify pharmaceutical agents for dark ion channels. Hopefully today I showed enough evidence that many ion channels are involved in human diseases, and CFTR was highlighted as one example that has a drug now, but how many other ion channels, specifically dark ones, have no drugs? So we need to find drugs specific against dark ion channels, and we believe this will have a great impact on human health. To develop these small molecules, we teamed up with David Weaver at Vanderbilt. We chose to focus on silent potassium ion channels. It is worth mentioning that potassium channels in general comprise the largest and most diverse class of channels that covers about a third of IDG list, out of which 23, highlighted here in purple, are pore-forming, meaning that they can form an ion channel, functional ion channel on their own, and 6 are considered auxiliary or modulatory, and those are shown in green. And among potassium channels, silent potassium channels represent a unique and poorly characterized subclass. What is so unique about silent potassium channels? So those channels don't really form ion channels on their own, but rather form heteromeric channels with Kv 2.1 alpha subunits, which, as you can see, allows immense structural, functional, and potentially pharmacological diversity. However, these silent Ks have been really poorly understood and characterized. We still don't really know what they do and where are they expressed. And because of that, there's no specific pharmacological or genetic probe developed against them. Together with Dave's group, we developed a system for high-throughput chemical screen for silent potassium channels. In this system, first, cell lines that overexpress either alpha or beta subunits of interest are incubated with fluorescent thallium indicator. Then, thallium itself is added to cells, which acts as an ion analog that can only enter the cells through ion channels. Once inside the cell, thallium is chelated by this fluorescent dye indicator that results in an increase in fluorescence, which is proportional to ion channel activity. The fluorescence is then read out by multi-well fluorescent plate reader. As you can see, it's a pretty simple setup. We can then couple this assay with an addition of a small molecule library to find ligands specific for ion channels of interest. For our first screen, we used a library of 50,000 small molecule compounds that were available at Vanderbilt. Those compounds were individually tested against Kv 2.1 cell line alone or cells overexpressing Kv 2.1 in addition to Kv 9.1. We are currently selecting the most compelling hits to be validated, but have already found a few putative activators and inhibitors, some of which are only active in the presence of Kv 9.1, indicating subunit selectivity. Shown on the right is an example of an activator that activates the dual cell line Kv 2.1 and Kv 9.1 quite robustly, but not Kv 2.1 alone. Currently, this and other hits are being validated individually in automated electrophysiological studies, and once validated, can represent very promising therapeutic agents for diseases in which Kv 9.1 is involved. Hopefully, I've convinced you today that ion channels, and specifically dark ion channels, are relevant for human health. I implore you to not just study well-known genes such as CFTR, but also focus on dark genes as well, since it is a wonderful area to make groundbreaking discoveries. And our center would like to offer help by providing data and resources for these dark channels. And if you found something interesting during this talk or want to collaborate, please reach out to me and my colleagues. Today I gave a broad overview of only some of the activities that we have at our center, but we have other projects with amazing collaborators that I didn't have time to talk to you about today. I would like to highlight Nevin Krogan and Ruth Huttenheim and our collaboration on proximity labeling of ion channels and their interacting partners, as well as Ashish Manglik, Dan Miner, and Gene Wells for our peptide and nanobody development against ion channels. I would also like to highlight our collaboration with Alan Busbaum, who is an expert in pain studies and is really interested in the role of some of these dark ion channels in chronic pain. And of course, Lily Jan, who is a world-class expert on ion channels and has been a very important contributor to all of these efforts. And lastly, I would like to give a special shout out to Zicheng Hu, a brilliant bioinformatician who's been extremely helpful in data mining, data analysis, and pretty much every aspect of all of these projects. Thank you all.